Hi everyone, this is my presentation on the dynamic pigmentary and structural colorations in cephalopod chromatophore organs. So basically the blue-ringed octopus and other cephalopods can have these incredible colors that can change rapidly. So how do they do it? So basically how they do it is they use chromatophore organs, and that allows them to change color. So these chromatophore organs are found in the skin of cephalopods. Some key features would be the pigment granules, as you can see in red, as they're pointed out there. Um, those are what provide the color. So the different colors that the octopus can be. The radial muscles as well, they're crucial in contracting, relaxing, and adjusting these pigment granules and the size of the pigment sac in general to produce different colors and stuff like that. Um, so what these radial muscles do is they pull the sac, they manipulate the shape of it, and it causes this pigment to expand or contract, and then it alters whatever the amount of color is produced. And these radial muscles, they run on electrical activity that can be found in the nerves. Um, and so, yeah, so whenever there's more electrical nerve activity, the pigment sac is widened, producing more color, etc. And then on the bottom, just below these radial muscle fibers, the kind of lumps, these are called sheath cells, which we'll talk about that when we move on later. So why are chromatophores important, and why are they so valuable to cephalopods? So the main reason, of course, I think a lot of people know this, would be camouflage. You know, it can be a huge tactical advantage for protection, for hunting, for a whole variety of things. So I say here, like hunting tactics and the waiting game, which is just basically how the octopus can wait for prey to come by, and then bang, they come after it. And then iridescence, which is a process which is can be a little hard to describe, but it's where based on whatever angle you look at something, it changes color, which can hide the organism even more. So here's an example of iridescence with a CD with a disc. Is when you hold the disc and when you like move it, you see different colors. You never like totally see the same color. So moving forward, as we can see here, so these are like different chromatocytes. They're labeled. So the ones that the arrows are pointing to are the yellow chromatocytes, and then the brown lumps, they're the brown chromatocytes. Um, so yeah, there's usually yellow, red, and brown chromatocytes that can be found in cephalopods. And so the first image is under one light intensity, and the other is a different light intensity. And these are different lighting angles that express different iridescence. So as you can see on the image on the left, their yellow chromatophores are a lot darker. They're a little bit smaller as well. And then on the right-hand side in image C, they're more reflective, they're more bright. And they may even be a little bit bigger as well. And then obviously in the middle, you can see the brown chromatophore on the left image is quite small, it's darker, as opposed to the one on the right, which is a lot larger and then a lot lighter. So these chromatophores have been manipulated by the nervous system to change their shape, which changes their translucency, reflectivity, or opacity. So this is, the basic knowledge of chromatophores was there. People understood what their function was and why they were important, but the in-depth nature that is needed to expand and move forward with science wasn't fully there. So they say here the complexities of the chromatophore organs weren't really known. One of the ways this could be done was examining it under a biochemical perspective. So prior to this research, chromatophore organs were mainly identified as pigmentary and absorptive organs. So the entire aim of this experiment was to just delve deeper and understand the dynamic pigmentary and structural coloration within cephalopod chromatophore organs. So how do we do this? So basically, individual chromatophores were isolated by removing sections of dermis from the palate, which is just a species of cephalopod. These samples were washed 5 times with filtered seawater and placed in a solution of collagenase. They're allowed to sit there for a while also. And then basically, simple as the individual chromatophores were plucked out using a micropipette and were separated by color. So as you can see on the right here, and the top image on the right, there's this whole image is the dermis, and you have different layers within the dermis. So top one chromatophore, and you go down, you get iridophores and lucifores. So we were trying to focus on the chromatophores, and so you can imagine you just kind of pluck them out one by one, and then they're separated by color. They're quite easy to tell by color, as we've seen in the other images between the yellow and the brown ones. And same thing goes for the red ones. And then more of the biochemical aspect was where we isolated the proteins from the chromatophores. They're purified and they were analyzed using mass spectrometry. And then these proteins that were separated were identified using BLAST, which is just basically a large database used to identify proteins. I'm sure a lot of you have used it before. And then all of this data was obtained and then analyzed to get the results that we got in this experiment. So the results. So basically, these organs are far more complex than we first thought. The chromatophore organs are structurally reflective at select viewing angles, which represents the iridescence that we talked about, just like the disc as you move around the disc. And then this structural coloration and pigmentary coloration is intertwined, making it a far more complex but effective process. So basically what this says is that everything's working together for cephalopods to, produce beautiful colors like this, but these beautiful colors they have more functions than just being cool colors. There's a lot of functionality to these chromatophore organs. So, like I said before, they're originally thought to just be exclusively absorptive and pigmentary, but not anymore. So structural coloration, which was one of the results that was determined from this experiment. So what is it? It's basically a mode of reflection in the chromatophore. So it can be formed based off structural components of the organism, such as the orientation as seen in the image. As you can see here, the different levels. You see the butterfly, we see one color, but it gets darker as there's different levels to it, etc. So different depths and different locations of particles can cause these different colors to be expressed. And then a direct optical observation of chromatophore iridescence was seen in the experiment that led to this conclusion. Essentially, that's structural coloration. Further results was that it was discovered that reflectant proteins surround the chromatocyte and are located in sheath cells, which are those little lumps that are found under the radial muscles. And it was also found that omega crystallin is the most abundant protein found in the chromatophore. So these sheath cells that we talked about before, they play a crucial role in these cephalopod coloration as biochemical components, such as reflectant proteins found in sheath cells. They're very important to this entire process of coloration. So as you can see in the slide here, this is referring to the proteins. This is more the biochemical aspect, because before it was thought that reflectin was the most common protein found in the chromatophores. But the proteins were sampled, they were blasted, etc. And it was determined that crystallin is the most common protein found here. And then on the left, on the far left of the graph, you see metabolism, which is the most, it's the highest, has the highest peptide count. And then you see reflectin towards the end. It's 1, 2, 3 from the right. There's a big difference. Crystallin is far more present there. It was also determined that there are more forms of reflection, that there are in fact 15, which is just goes to show the complexity of the chromatophore organ. So what is omega crystalline? So this protein has two main roles. It contributes to the nanostructure of the granule itself, and it contributes to the stabilization of xanthomatin in the granule. So basically, it maintains the structural integrity, which is crucial because that's the source of the color, basically, and the pigment. And then the need to stabilize this XA. Why do you need to stabilize it? Well, this xanthomatin is a toxic byproduct of tryptophan metabolism, and it has toxins in it, and it can damage the organism. And so this omega crystalline has two major pigment binding pockets on the left and right regions, which can be seen here. A is the left, B is the right, they're much less selective than the pockets on the reflectin proteins. So as a result, the XA, they have a more favorable interaction with the omega crystalline. So basically, overall, both features, they are key contributors to pigment color retention, light absorption, scattering as a chromatophore experience a change in presented surface area during actuation. So how does this affect me? What's the point in this? I think that's what I was trying to get at. So the human cephalopod, how can we even be remotely similar? Well, we can. So basically, humans have homologs of receptors that mediate pigment translocation in melanophores. And melanophores are types of chromatophores that contain melanin, which is, it's obviously in our skin. And those are the kind of most relatable types of chromatophores to humans. And so the similarities between this aspect of our morphology and physiology allows scientists to take advantage of this. So it enables us to use chromatophores to model certain diseases, and then to also design drugs for things such as appetite suppression and tanning. So the future applications of this. So how can our research that basically discovered the complexity of chromatophores, how can it be used in the future? So scientists have been able to use melanophores as biosensors to detect certain diseases that are mainly associated with pigments, such as melanoma and albinism. And it was also recently discovered in a separate study that the gene responsible for the melanophore-specific golden zebrafish was shown to have a human equivalent that strongly correlates with skin color. So we're not that different to cephalopods, I guess. So additional use of this, basically, they're pretty cool. Cephalopods are very cool. Chromatophores are very cool. How they work is very cool. But another use of this would be in the army or military. So the chromatophore mediated color changes that can act as a type of dynamic camouflage for soldiers. So just like the octopus, so it's like an octopus, for example, can change color like that. What if a soldier could do it like that? And that's one of the, I guess, next step in chromatophore and cephalopod research and laying the groundwork in this study. So, with the knowledge obtained from this research, it also, it allows us to understand the complexities of the chromatophore, affording us more avenues to conduct research and start new projects, surrounding chromatophores and overall how they can impact humans in the future. Thank you.